very happy that Kyle has agreed to take the comm, as it were, for our focus group, where we really want to talk about the fact that every time I go to any webinar and I present, I hear people say, so when are you guys going to represent single cell results, particularly in disease states and non, on the portal in the context of human genetics? And I say, we're really working on that. But the challenge with that is the fact that there are really beautiful data generation activities going on. There are means to process those data, and there are means to represent and visualize those. But then how do you, and there's many resources that are still being built, you know, that Kyle will probably mention when he talks, to represent them. But then how do you then pull that together with what we know about human genetics? So today we're going to talk a little bit about that. So how would you use, you know, single cell results from, say, kidney disease or non-kidney disease, or pancreas samples with, you know, diabetes without, in the context of human genetics, because that's really where we start from. So I wanted to sort of give you some context of where we're starting from and then hand it over to Kyle. And again, nobody's muted. You can unmute yourself, you can chat, you can raise your hand. This is very informal, meant to really be about discussing the future of how we visualize in the portal. So I'm just going to quickly show you kind of where we stand today on the portal, how we think about, you know, representation, because it's extremely new, right? So if you go to the common metabolic disease portal, you can search by a gene, a region, a variant, or phenotype, right? Those are the classic entities by which we represent data. And you could do this across disease-specific portals if you don't want to go across the entire resource. These are our, you know, well-known portals. But what, you know, increasingly we're trying to understand is, you know, for the tissues that are relevant to diseases, what's happening in them, because we need to understand that, you know, the molecular mechanism, the pathway, the function, and whatnot. So trying to connect the tissue, the cell state, with the phenotype and with the observation in the patient is a challenge. So we started with a very, you know, an early version of that on our gene page, where if you search your favorite gene, you know, you'll get the human genetics evidence summarized across common and rare variation for you with what we call our huge score. You know, it's going to give you the bottom line evidence, if you will. And then down below, we start to sort of layer in, okay, well, if you want to, you know about the human genetics, what about, you know, that gene's expression, right? That's useful, but that only gets you one way. So right now we represent, you know, just mRNA bulk expression levels across the tissues we have in CMDGA based on a set of collections. And what's lovely about those collections is they come from two broad resources. One is, you know, the disease agnostic resources like ENCODE, Blueprint, and GTEx. But moreover, and increasingly in our field, we have really elegant resources like HPAP and KPMP, the Kidney Precision Medicine Platform, that are generating, you know, both bulk and single cell and nucleotide level data sets in disease and non-disease states. So how do we represent that on the portal? How do we make those accessible and integrate those? So we've done a first blush, which is very quick, just to give you an idea that we're starting to bring those in, right? So right now on the portal, you can see KPMP data for bulk data sets, and you can visualize where they are, but you can't say anything about the single cell level, right? You can see the expression level, you can drill down to the exact sample in CMDGA and go to it. So that's awesome, but we want to bring you to the next level. So with that today, we're really going to frame that discussion. I hopefully I gave Kyle a good intro. He and I kind of talked beforehand, but I'm going to turn it over to him. And again, pop questions in the chat, and we're going to really talk about how to visualize these data and how we're going to aggregate them and work with our collaborators to make these more accessible broadly. And I will stop share, Kyle.
Okay, thanks, Noel. Can you hear me?
Yes.
All righty. Okay, so yeah, so thanks, Noel. As Noel said, I'll be talking today a bit about single cell data, how we provide maps generated from single cell data, resources that can be derived from these maps, and then, as Noel said, thinking about how these data can best be visualized in the portal, and how we can provide people insights from these data. So please feel free to interrupt at any point or ask questions. I'm not sure how these focus groups typically go, but you know, it's fine with me if people want to interrupt at any point to ask questions. And then I also have at the end several questions that I've outlined that we can discuss as well after I'm done. Okay, so single cell data. So I presume most people have some basic understanding of single cell genomics, but if not, single cell data enables you to take a heterogeneous tissue and then derive gene regulatory programs in individual cells. Okay. As well as individual cell types from that tissue. So on the left here, I have just a very basic overview of what happens to go from generating the data, where you isolate cells or nuclei, you perform the single cell assay, you generate profiles, bless you, that look.
Sorry.
Oh, it's all right. That look like this, basically, where you have individual cells on one axis, and then if it's gene expression data, genes, if it's chromatin data, genomic regions on the other axis, where you have the profiles for genes or these regions for each individual cell. But oftentimes that by itself is not particularly useful because you don't know, for example, what cell type a cell arises from, and so forth. So there's a fair amount of processing and then clustering of these cell profiles that then resolves the cell type of origin for these cells, which you can then use to derive profiles for each of these cell types. So what types of information on the right here are being derived? So we have gene expression data, right? So the expression level of genes within a cell type, which can be profiled using single cell RNA-seq or a single nucleus RNA-seq. And then commonly as well, people do assays to derive the location of cis-regulatory elements, which are regions that are driving the expression of genes in cell types. And so these cis-regulatory elements or CREs can be assayed using assays such as a single nucleus attack-seq. Increasingly, and I'm sure a lot of people in the consortium are generating these types of data, multi-omic data, which is paired profiles where you get both RNA-seq and attack-seq from the same cell. It's also hundreds of different types of single cell assays, many of which were reviewed in this paper that was from myself and Sebastian Pressel. He used to work at UCSD. And then also Bing Ren in Nature Reviews Genetics, where it's summarizing all the different types of assays that have been developed to generate single cell profiles, and then also what pieces of information you can derive from different types of assays. But what I've listed here are the most commonly done assays, and probably those people are most familiar with. Okay, so so that's single cell assays, and then what you can derive from them. And again, you can derive cell type profiles, so understanding gene regulation, individual cell types, as well as you can look at the individual cell level to understand how gene regulation is. Yeah, okay, all right. Klaus already. So okay, so so Klaus's point is correct. So I should have highlighted that. So I called these cis-regulatory elements, technically speaking. Attack-seq identifies candidate cis-regulatory elements because in most cases those haven't been proven to regulate gene expression. So we tend to call these candidate cis-regulatory elements or C-CREs, demonstrating that a region actually drives expression of a gene is a bit more work in many cases, or requires different types of data, right? So attack-seq is finding regions that are really demarcating where regulatory elements might be. But then again, these are often just candidate elements that require, depending on who you ask, probably Klaus included, functional validation. So that is an important point to distinguish. Okay, so I'm going to talk a bit about resources that are ongoing to provide single cell genomics data to the community, in particular resources that generate data that's useful for understanding metabolic disease. So one resource that is relevant to Klaus, because he runs this program, is called HPAP, which generates data from the human pancreas, purified pancreatic islets, data generated by HPAP on human donors that could be non-diabetic donors, as well as type 2 diabetic, type 1 diabetics, a range of disease states relevant to the pancreas. the data generated by this program is deposited in this database PankDB. And then we have, and then also Klaus and his colleagues as well, that take these data and then derive single cell maps from these data. And so, for example, on the right here, this was a paper we published a couple months ago in diabetes, where we took data from PankDB from single cell gene expression and then created an integrated map of cells from the pancreas, where you identify cell types in islets as well as other cell types in the pancreas. Just a quick aside about what we're looking at here on these maps, right? So these maps are, each dot is an individual cell, and then they're colored based on the cell type that was assigned to the cluster that these cells are grouped into. So the clustering is automated, it's computational, so it finds these groups of cells. But then after clustering, you're left with just groups of cells that you then have to annotate what the identity of these you believe these cell types is. Most often that's done by looking at the expression of genes that are known to demarcate certain cell types. So, for example, in beta cells, they express insulin and other markers, and so the cluster that highly expresses insulin we would annotate as beta cells. Glucagon is the hormone produced by alpha cells, so cells highly producing glucagon we annotate as alpha cells, and so forth. So, that process oftentimes is manual, but looking at the expression of different marker genes across different clusters, it's also possible to do that in an automated fashion to annotate cell type identity. But we and I presume many others do the identification of cell types still based on kind of manual assignment based on expression of key marker genes. And then, so these maps then, as I said, contain data from many donors. In this case, 65 non-diabetic, type 1 diabetic, and type 2 diabetic donors. And so one key thing in taking these single cell data and producing these maps is the integration of profiles across donors. I won't get into the details of how that's done, but essentially it's some level of correction for batch effects across different variables, and there's different thoughts about the best ways to do that. But ideally, in the end, when you cluster your profiles and account for batch effects, say donors or the source of the tissue or so forth, then the representation of cells for a given cell type is fairly uniform across those axes. So if we look here in this map, for example, in beta cells or alpha cells or other cell types, and we color the cells by the donor that they come from, there's fairly even integration and representation of each of these clusters from different donors. On the top right here, similarly, HPAP has data that I think, well, I guess they're all generated at Penn, but the tissue comes from either Penn or from the University of Florida. So the source of the tissue is a potential confounder. And so, but by considering batch effects, you could see that the source of the tissue is also fairly well integrated across these clusters, right? So when you talk about integrated maps combining data across single cell assays, that's what people are referring to is integration of data across many donors that could have different properties and sources, biological states, and so forth. All right, so HPAP is one major resource, and we've done a lot to aggregate data from HPAP. We worked with Klaus and PankDB for many years to take data that they generate and try to create maps from it to provide to the community within the portal. Another resource I just want to highlight that Noel also mentioned is KPMP, which is an NIH-funded program to generate data from human kidney donors. There's a website here where KPMP provides the data that they generate, similar to HPAP, but for diabetes, that they generate in KPMP data from controlled non-kidney disease donors, as well as donors that had various types of kidney disease. And you can look to see what the representation of different disease states is in this database. In this case here, so the KPMP consortium had already made integrated maps of all the cell types in the kidney. from data that was deposited in KPMP. We were and had several communications with them about the data that they and resources that they generate. So we took the maps that they had generated themselves from KPMP, which has a pretty large diversity of different cell types and subtypes in the kidney in this map, and then took information from that to display in the portal. So those are kind of tissue-specific resources, right? Pancreas and kidney in areas that are of high value to metabolic disease. I'd say there's also pan-tissue resources, so resources that aren't necessarily focused on a specific tissue, but they generate data across different organs or tissues, and they don't have necessarily an interest in a particular disease area, but they generate data across a wide breadth of tissues that's useful for a large range of diseases. So there's several of these resources, one of which is called Tabula Sapiens, and then the data from Tabula Sapiens is hosted, I think, by Chan Zuckerberg, but there's a portal for Tabula Sapiens, and they generated data from 24 tissues. And then there's also the Broad single cell portal, and then Chan Zuckerberg has its own single cell portal as well, where they provide maps from different tissues, right? So all these data, and is quite a lot of data, particularly in the Broad CZI portals, that is useful for understanding gene regulation and many different tissues across the body. So that's gene expression, and then I just want to briefly touch on chromatin as well. So there's also an increasingly large number of single cell attack-seq maps that are being generated. Again, can be tissue specific. So we several years ago published a study looking at single cell attack-seq in pancreas and blood, and the reference is here. And then also Bing Ran's lab and us, and this was a project actually that was funded by the FNIH in the last version of AMP, to generate single cell attack-seq data across a large number of human tissues, and in total identified 222 different cell types, and derived chromatin profiles in each of these cell types, and that was published in Cell a couple of years ago. Okay, so so those are the starting point resources, I would say, which are integrated maps of either gene expression or chromatin profiles. And again, those could be from tissue-specific resources that have a particular focus, or those could be pan resources that generate data across a wide range of tissues. So from here, then the question is, what types of knowledge can be derived from these maps, from single-cell genomics data that's useful for understanding gene function or understanding in the context of the portal. So given an integrated map here of cell types, here are some of the things, but certainly not all, but some of the pieces that can be derived from these maps. So for example, things like marker genes of particular cell types. Those could be marker genes that we already know about. For example, insulin in beta cells is not is an obvious marker gene of beta cells, but there may be other highly specific markers of cell types that haven't been identified yet, or if there's a gene of interest that somebody's studying, may have highly specific expression to one cell type relative to others. So we can derive information about which genes are highly preferentially expressed in one cell type relative to others. Two, derive the expression of all genes across the genome in every cell type, right? Most genes aren't expressed specifically in a cell type relative to others. That's a very small set of genes usually. Most genes have some degree of expression in many cell types, and but to varying levels, right? So to understand the expression of genes within each individual cell type across the genome. Similarly, what are the cis regulatory elements that are active in a given cell type across the genome? What's their activity across different cell types? And then also, what are the genes that are likely regulated by those cis regulatory elements in each cell type? Four, what are the changes in expression of genes or cis regulatory elements across a phenotype or disease state? Yeah, that could be things like diseases, but could also be normal phenotypes that like physiological parameters that everybody has, such as age, BMI, things like that. Other things that can be derived from these data are some insight into heterogeneity. So if there are subtypes or substates of cell types, some of which may not have been previously known before these experiments were done and these maps were created. So for example, in the pancreas, I think probably not too controversial at this point that there have been identified, just for one example, multiple subtypes of ductal cells, in particular a subtype of ductal cells that has high expression of mucin genes. And so, these subtypes of ductal cells were identified from these single cell maps where these cells cluster differently than normal ductal cells and also have slightly different gene expression profiles. And maybe those subtypes have different roles in disease, different changes across phenotypes and so forth. Single cell data also allows you to look at dynamics, right? Like, so continuous dynamics, like trajectories across disease states, things that people have probably heard of, such as like pseudo time, where you can order cells along an axis. Doesn't have to be time, could be pseudo disease or pseudo phenotype. Like, it could be any axis that you want to order cells along. Since you have individual cell profiles for a cell type, you can order cells along an axis of interest to see how the dynamics of expression change across that dimension. And again, there are more things that these data could be used for, but these are some of the key things. Okay, so all right, okay, so I'll talk for a minute about back to yeah. Somebody say something.
Was there a question? No, I think you're good, Kyle. Keep going.
So back to a second, the HPAP maps that we made. And so again, these are data from the human pancreas, from PancDB. And what was the chat question here? Oh, okay. Okay. So the things that can be derived from these maps. So as using this HPAP data as an example, we have definitions of every cell type. And then from these cell types, we can derive levels of expression of genes within the cell type. And you could do that across the entire set of samples that you have, right? So you get one value across the entire cell type, but more often you're probably interested in understanding the expression of a gene in a cell type within each individual sample in your map, right? So so to do this, we create normalized gene expression profiles of aggregating cells within a cell type per sample, so that you have sample-specific gene expression levels for each cell type. So this is just an example in beta cells of some key marker genes of beta cells, where we have the normalized expression of each of these genes in beta cells, and each dot here is a different sample, right? So, and these are non-diabetic individuals. And for the most part, there's fairly consistent expression of a lot of these genes across donors, right? But this is a key piece of information that's derived from these assays, and that's represented in the portal at the moment, which is sample level aggregated profiles for each cell type. Other things that can be derived from these maps are changes in disease, right? So again, using HPAP data, we looked at changes in gene expression in each cell type in disease states. So there are type 1 diabetic and type 2 diabetic donors in these data sets. So just to give you one example, in beta cells in type 1 diabetes, if you ask what genes within beta cells have changes in expression across samples in type 1 diabetes compared to control, this is what the results look like. So we find upregulation on the right here of many genes that are a class 1 MHC. So this is antigen presentation genes, as well as genes that are involved in helping class 1 MHC present antigens, as well as cytokine response factors, right? So this gives you pretty decent insight into changes that have occurred in beta cells and in type 1 diabetes compared to non-diabetic donors, that they're upregulating these cytokine response and antigen presentation genes. And this is just to show that if you look at the pathway level, if you say what pathways are these up and down regulated genes enriched in at the upregulated gene level, things I just said, antigen presentation, cytokine signaling type pathways come up highly enriched in these sets of upregulated genes. So that's what we can derive from gene expression maps, levels of expression in cell types, changes in cell type expression and disease. From chromatin maps, probably the key thing that's derived from these maps is once you have the clusters and the cell type identity, then deriving the location of candidate cis-regulatory elements for each cell type, right? And so on the right here, this is showing tracks which represent each of the different cell types, which is on the left here, and the chromatin profiles for each cell type, where cis-regulatory elements are defined individually for each cell type, right? So you get definitions of where these regulatory elements are located in each individual cell type. Similarly, in the study that from Bing's lab, where they looked at pan tissues, they derived cis-regulatory element definitions in each cell type, and as I just mentioned in the previous slide, they further generated predictions within each cell type of the genes that are regulated by these cis-regulatory elements using a technique called ABC, which leverages ATAC-seq profiles from each individual cell type, right? So this study not only derived the locations of these candidate cis-regulatory elements, but also the predictions of genes that are likely regulated by these cis-regulatory elements. Okay, so all right, so then I'll talk a bit about now how some of this information is currently displayed in the portal, and then a bit about some ways moving forward that we can think about to enhance basically the way that these data are shown. So there's a handful of ways at the moment that results of single cell assays are displayed in the portal. Just to note, at the moment, everything that's shown in the portal is what I'm calling an aggregated cell type profile, right? So that's something like aggregation of expression of a cell type at the sample level, or aggregation of cis-regulatory elements at the cell type level. I think, and Noel touched on this a little bit, I think part of what we want to move to over time is not only showing these aggregated profiles, but then also allowing people to look more in depth at individual profiles and how, visualizations of individual cell profiles might enhance your interpretation of some of these data. But at the moment, everything shown in the portal is something that's aggregated at the cell type and sample level. Okay, so what are some questions that you could ask from these types of results? So Noel mentioned this, but I'll just reiterate. So one question is, where is my gene of interest expressed, right? So if you go to the gene page and then search for the gene, and then a module on the gene page shows you information about expression of your gene in across different tissues. Some of this is bulk data, but then increasingly some of this is single cell data. The way that this is shown is that it's aggregated at the tissue level, but then if you hover over the tissue, it breaks it down by the individual studies that are contributing to that tissue. And some of those are data from single cell studies. So for example, if we look at the CTRB2 gene, which is an enzyme produced by acinar cells in the pancreas, and you hover over pancreas, at the top here, you see that the highest expression of this gene is in pancreatic acinar cells. If we wanted to focus in on data from just a specific source, so for example, HPAP, this drop-down menu here, collection, lets you select a specific source of data. So if you select HPAP, then that just shows you pancreas to start, because all the data in HPAP is in pancreas. And then again, you can hover over and see the different expression of samples for different cell types. you can then on the bottom here expand this out to see the summarized expression levels of each individual cell type from this HPAP data in the pancreas. And again, this is data is aggregated at the cell type level per sample, and it shows you information about where these genes are expressed in each cell type. And you can see pretty clearly that if you look at the normalized expression values, that it's expressed much more highly in acinar cells than any other cell type. It also provides links to go to where these data actually are if you wanted to download these results and look at them further yourself. Here's another example, this gene NKG7, which is a marker gene of NK cells, I believe, in the kidney. So if you want to look at this gene and know where it's expressed in the kidney, you can select this KPMP collection, and then again, hover over the different dots here, which are different samples grouped by cell type within the kidney. It's most highly expressed in NK cells. And then again, if you expand this menu on the bottom and look at each individual cell type, there's no room to show all this here, because in the kidney, there is an incredibly large number of cell types and subtypes that were identified. So you'd have to scroll down pretty far to access where the NK cells are, but it's pretty specifically expressed in NK cells. One thing, as I was putting this together last night, one thing I noticed is that, well, so sometimes we have multiple representations of the same biosample, so you can see it right here. And I think this is actually, since we derive gene expression levels separately for different disease states, this is probably representing different disease states, although I don't think this is actually annotated on this table at the moment. So that's probably one easy way to give people insight into not only what cell types these genes are expressed in these key tissues, but then also what's the expression in the cell type in different disease states. Okay, so that's how you can look at expression of individual genes. Another question that is of interest, of course, is what genes are associated with my phenotype disease state of interest. So at the moment in the portal, the way that access this information is through what's called the Screen2 gene, which is accessible from the homepage of the portal. There are in this, this is basically collections of genes. It could be differentially expressed genes. Some of these results are like the results of functional screens. If you want to look at differentially expressed data, you can go to the data type menu and just select differential expression, and then that shows you a list of differentially expressed genes. So at the moment, a lot of the differentially expressed gene lists are from the data sets I described, KPMP, the kidney data set, and genes with differential expression in kidney disease phenotypes within each cell type, as well as data from the HPAP, which is changes in pancreatic cell type expression in diabetes. So if you click on one of these, I'm showing here the beta cells in type 1 diabetes, which I showed earlier. This gives you a high-level overview of the differential expression. So a volcano plot of showing the genes with most significant changes in expression and their full change in expression, in this case, in type 1 diabetes and beta cells. At the moment, I think this just shows the significant genes. So that may be also something to think about, Noel. Yeah, well, cite the, what do you mean, Klaus? I mean, the papers are cited. I mean, yeah, I guess this.
I mean, there've been two papers which are the HPAP, describe the HPAP program. That's where the data come from. So therefore, they should, of course, be cited on the knowledge base. I
think this is, so that's a good point. And that's something that we can maybe get to a bit later, is that I think, Noel, there's really no pages that summarize the resources that are providing these data, right? At the moment. So, and actually, I was thinking that would be also a good place to put, if we wanted to display the integrated single cell maps or something, right? If you had a page for HPAP, that might be a good place to put things like that. And then that would also be a landing page to highlight like the citations for the resources and stuff like this, right?
Yeah, I think the resource is not clear here, but you're right. There's like the paper is up in the corner that it came from, but the resources, I love that idea, actually. That's a really cool thing we could integrate.
I mean, the paper cites the resource, but,
yes, but like, the resource should be cited.
I think it's a separate thing, right? Like these results are generated by paper, which may itself use other resources, right? But then I think the resources themselves probably need some place to highlight, like like the KPMP or HPAP, like to highlight the overall resource, right? And then again, that could actually be a place where like the integrated maps derived from those resources are shown, because I think this otherwise is not actually an obvious place to put maps like that in the portal, right?
Yep. That's a really good point. I like that. Klaus, does that get at your point as well? Yeah.
Yeah, I think.
Yes, indeed, absolutely. I mean, to me, the data generators have to be acknowledged here, not people who took the Sora object and process it further. So
yeah, absolutely.
Here as well. Thank you.
I like that.
Yeah. I think that's, but I see those as two different things, right? Like one is like the resource, and I think those probably need a separate place to describe the resource and things, like made from the resource that use data only from that resource, and then things that people may derive from that resource. It can maybe refer back to that page as well. Probably is the way to do it. All right. So okay. And then, and then you can also look at individual genes. You can also filter by gene of interest. So if you're interested in knowing whether your favorite gene has changes in expression, then you can look that up as well. Okay, so that's so those are two ways that you can look at gene expression data: expression of a gene on the gene page, and then also changes in expression associated with a phenotype. Okay, so some other things I wanted to describe in the portal are more related to the use of cis regulatory elements, right? So gene expression is useful for understanding, obviously, the function of a gene and where it may be acting and how it may change across a phenotype. Cis regulatory elements have that property as well, but they also have the property that you can intersect them with genetic variants that are associated with disease. And so you can use cis regulatory elements to gain insight into how they relate to where disease-associated variation are falling. And so one way that's commonly done to do that is by asking whether variants collectively associated with a given phenotype are enriched in regulatory elements active in a given cell type or tissue. And so on the phenotype page, like if you search for type 1 diabetes or AFib or something, one of the things that's shown is the extent of enrichment for variants associated with that trait for regulatory elements in different at the moment tissues. So I don't know what the timing is for the cell type resolved version of this, Noel, but at the moment this is collapsing the information down to a tissue level. I think that the plan is to show these types of results at the individual cell type level. So for example, like for AFib, right, we're seeing enrichment of cardiovascular system or heart. If you separated that out by cell type, you'd see enrichment, I think, specifically in like cardiomyocytes and maybe a lesser degree to other cell types, right? So breaking that result down, but I mean, I don't know if Jason's on, but I know that's the those results are either exist or being generated to put in the portal, but the cell type separation of these results, right, then gives you insight into which cell types are probably most related to which traits based on variants associated with the trait overlapping elements active in that cell type.
I agree. So I think Jason's on, but I think that is sort of one way we're thinking of doing this. I know DK's on, because I know he's working on that from the tissue perspective page. But I guess my question was, how, since we're in a focus group, how best would be visualized, Kyle? Would it be just a table? Is there some sort of, like, is a UMAP plot at that point useful? Like, what would be the best way to present that? Because there's a lot of cell types, right? In many cases.
It's a good question. I think probably would need some level of grouping by tissue, so that you're not just dumping out every cell type in one list, right? Like what's done for gene expression, if it's organized by tissue, and then you can expand out the tissue to see like where they. I mean, I guess the issue there is that at the tissue level you may not see enrichment, but at the cell type level perhaps you would see enrichment, right? Like if there was a rare cell type that was enriched for disease, that may be not observed at the tissue level, right? Because it's a small proportion of the overall tissue. But that could be one way to do it, is group it by tissue and then expand it out by cell type. Yeah, I'm not sure. I mean, tables, I think in this context, I find useful, but
would it be like one of the things we're thinking about, or at least I've been thinking about, and maybe others, would be if you were to query by tissue, for example, like I think that's one thing we're thinking about, like an entry point that is specifically based on, tissue enrichment, and then allowing you to visualize by cell type under that model. Maybe you want to go beyond a table and have like, would the UMAP plots be valuable there? Would there be some sort of like interactive visualization that would be really useful to present, that's already either built that we could implement or that we could envision being useful to our user base?
To show like the high level enrichment?
Yeah, I don't like, is there a standard way that people represent these in papers for that particular type of visualization that we could leverage?
Usually people just show like heat map type things or bar plots or something, right? Which at some level may not be that much more valuable than a table, but
got it.
Others may have thoughts, but I mean, it's just an enrichment, I think, at this point, right? Yeah, I don't know. I mean, maybe others have thoughts on this, but yeah, I feel like a table probably serves a purpose pretty well. But you could show like a heat map or bar plots or just to show the extent of enrichment. I mean, I guess if you had a bar plot, you could maybe have a bar plot at the tissue level and expand that out to the cell type level. Maybe a way to make a visual that makes it a bit more intuitive for people than just a table. But I don't think there's anything like out of the box that I know of that could be a specific question. Yeah.
Would it be valuable then to see the side-by-side comparison of disease state versus non? Like, would the enrichment be a valuable thing to see across the same cell type?
Yeah, I think that same cell type in different disease states. Yeah, I think that could be valuable. Yeah. I mean, depends. Yeah, I don't know. Depends on I mean, most of the information in a cell type is probably not changed in disease.
That's probably true.
Depending on the disease you're looking at. So you like a subset of genes are changing or a subset of sites are changing. And like these types of analyses, since they look at the sites across the whole genome, if you like ask whether sites for non-disease or disease individuals show different enrichment, they probably would look fairly similar, I would think. But that there may be different types of analyses that get at that question, right? Like are the subset of sites that are changing in disease show preferential enrichment for a phenotype? That's a bit of a separate question than this is asking here, but that could be also a valuable thing to consider including. But yeah, I mean, maybe a visual that allows people to look at compare the magnitude of enrichment visually with the bar plots or things like that could be valuable. Yeah, and a way to filter that by. So I mean, you could do filtering by tissue, maybe by type of cell, right? Like if it's an immune cell, and maybe you could look at immune cells across different. Like there's probably multiple axes of how you'd want to filter cell types. One is like, what are the cell types within a tissue of interest? Another one is what are the cell types across like a lineage of interest, right? Like if you want to look at enrichment across different immune cell populations across different tissues, that may also be valuable in addition to looking at all the different types of cells that are within one tissue.
Got it. Cool.
Okay, so that gives you high level insight into the I do want to save some time for questions. Okay, I'm actually gonna. Well, is this an hour, Noelle?
Yes.
All right. Okay. So one other thing I just wanted to briefly mention here is that something that we've been working on is trying to, given that you may see multiple cell types that are enriched for a trait at a high level, you might want to know which loci are most relevant to which cell type for that trait. So one thing that we have been working a bit on is to try to summarize the information for individual loci associated with a disease per cell type, using these cell type cis regulatory elements, and then basically rank or give people a rank list of loci based on the probability that the variant for that locus falls in a site for that cell type. So for example, for type 1 diabetes, numerous cell types involved in type 1 diabetes, immune cells, other pancreatic cells. And if you summarize which loci likely affect T cells versus, say, acinar cells, you get very different sets of loci. And then, so if you're interested in studying a particular cell type for disease, this may give you loci of interest to pursue. Also, if you're studying a particular locus and you want to know like what cell type, there's some evidence that locus is preferentially affecting for that trait, it can tell you that as well. So, for example, for acinar cells, there's some loci that are where it's highly likely that the causal variant falls in a site active in acinar cells. And one example is shown here. All right, I'm going to skip forward a little bit just so I can talk about visualizing data and some new ways we might want to think about visualizing data, right? So in the portal, as I said, everything is kind of shown at an aggregated level. So aggregated cell regulatory elements, aggregated expressed genes, and so forth. And I think there may be some value in giving people the opportunity to look at expression more interactively, or chromatin more interactively at the individual cell level. So we do have some visualizations of individual cells in our CMDGA, which is the site that provides the genomic data to the portal. If you go to our site and look at this tools and cell browser, and then you go to cell browser, this and then you can look at the interactive analyses of the integrated maps for these different data that's included in the portal. So this is one example. So back to the HPAP data, right? So this is showing the integrated map of HPAP. And you can search for particular genes. And now instead of seeing where it's expressed in aggregate in a cell type, you can look at the expression of this gene in individual cells. So for CTRB2, again, enzyme expressed in acinar cells, highly expressed in acinar cells relative to other cell types, but there may be more nuance to questions like this, like maybe it's expressed in a gradient or something in a cell type, or certain parts of a cell type that may give you more information. Another example is MAFA, right? So this is a beta cell expressed gene, and you could say it's pretty specifically expressed in beta cells. But so displays like this may or may not have some value. I personally don't find these types of ways of looking at gene expression usually too informative, just because I'm not always sure exactly what they're telling you outside of it's expressed in that cell type, which you would already know from the aggregated data. But there are things that you can do at the individual cell level that may be a little more informative. So again, these visualizations allow you to then look at expression of a gene of interest per cell type, but instead of the dots being individual samples, now here the dots are individual cells. So these types of visualizations may give you some insight into, sets of cells may be expressing a gene, or other sets of cells may not be, questions like that. Also, can see patterns that are at the individual cell levels, is my point, that may be obscured at the sample level. You can also subset these by different phenotypes that are included. So disease state, right? So this is different diabetes disease states, but you can also separate based on sex or age or BMI or any other phenotypes that are included in these maps. So this is an interactive way to look at a gene of interest across any dimension that phenotypes that were collected for the studies that are in these integrated maps. So you could ask questions of interest yourself that may not be things that were necessarily derived and put on the portal. Yeah. So I think, right. So David, it would be great if we had some examples of, like, I don't personally have any great examples of where this has actually been useful for me, but if you guys do that, that would be fantastic. Because, yeah, I guess if you have subclusters of a cell type that you think are important, then looking at where the gene is expressed within that subtype, this could be a valuable way to look at it for sure. All right, so yeah, so then this lets you then separate the data on whatever axis you want. So it gives you a bit more control over looking at different dimensions, and you can combine different phenotypes, look at disease state, and sex and things like that. Okay. And then this is, yeah, just some insight into how you access the data, but I think that should be relatively straightforward. Okay, so I'm going to stop because I'm going to leave enough time for discussion. So, I think some questions that we're interested in getting some thoughts on from people are, first of all, to what extent is there interest in viewing these individual cell profiles, like questions like this, or questions like this, in the portal, in addition to looking at information aggregated per cell type? And then also, what questions would that help you address that can't be addressed now just by looking at aggregated cell type information? Two, are there modifications, additions to the current visualizations that you think could be valuable, and or what additional types of visualizations could be valuable to include? The last question I had was, are there other pages or analyses that people can think of that would benefit from including visualization summary of expression and these results outside of the places that they're currently included on the portal? So I'll stop here, and then yeah, if people have any thoughts or things they want to discuss, we can do that now.
Yeah, this is Klaus. So on the last point, I think you might consider incorporating, the cell type specific high-C maps that are generated recently, including from our lab, because you talk about candidate regulatory elements, which are open chromatin regions, but to be able to then say which of those links back to which promoter might be useful, especially when you're interested in identifying variant to gene interactions. So that's something might be difficult to incorporate, but something to consider for the future. Thank you.
Yeah, that is a good point. So there is, Noel, there is some of that types of information, right? But I don't know how useful it is at the moment.
Yeah, we have some of that now in simple tracks in the variant sifter.
You mean, Klaus, you mean like in your guys' study, that was bulk profiles per sorted cell type, right?
Right, but it was for alpha cells, beta cells, and arsenic cells, and it's very clear that it helped a great deal in prioritizing, signal to gene interact relationship and which cell type that might be more relevant.
Yeah, for sure. Yeah, no, I think that's, yeah. So I didn't know whether you meant that or like single cell high-C type data, which, yeah, visualizing that would be even more. Okay, so like aggregated cell type chromatin contacts. Yeah, I mean, so there is the data from your guys' paper is included. I think it's, Noel, correct me if I'm wrong, in these prioritization pages, right? Like if you try to prioritize variants that are at a given locus of interest, one of the pieces of information that you can include and separate variants on is what genes are linked to certain variants based on multiple pieces of information, ABC,
yep,
CISRO, which is like, or like, any like single cell correlation based method, and then also interactions from high-C based assays. Yeah.
Right.
But there's no visualization of that, right? It's just like,
no, it's just a track at this point. Yeah.
You just select the types of data of interest to you, and it just shows you the variants that satisfy those criteria, right? And the genes that they're linked to.
Yeah, the overlapping. And then you could like click out to CMTGA though to actually see the data, but again, not a full-fledged visualization.
Well, one thing we don't really at the moment have like a chromatin or like a genome browser that shows this type of data, right? Like the profiles per cell type. And we kind of did in LocusZoom, but then I noticed it's not there anymore on the Locus page, right?
Yeah, it's on the variant sifter, right, which is specifically a tool for variant prioritization, but allows you to visualize those overlapping maps in sort of a browser window because it appears below your LocusZoom plot and aligns with, credible sets and other variants, but it's really for variant prioritization. But we could consider, how to make that more useful in the context of visualizing these annotations rather than variant prioritization per se.
I mean, I think this is very structured and guided, right? Where you're like picking things and then like it's navigating you through like a process. The endpoint is like list of variants.
Yes.
Satisfy the criteria in genes. I guess there's a orthogonal, like just look at a region of the genome and see like here are the, say you want to see the tracks for all the cell types for different islet cell types, and then the loops, right? And like an IGV type browser that shows you all that information together, and you just want to peruse a locus in that way is not that straightforward at the moment, I think, right?
This is a good point. Yeah.
Can I ask a question?
Sure.
Yeah. This really.
Go ahead. Go ahead. Yeah, go ahead.
This really fabulous talk. I really love it. For the last part, when you mentioned that aggregated cell type, when you use a gene expression to prioritize like which GWAS locus enrich in which cell type, I wonder whether you can do the similar thing for the open chromatin using ataxic data instead of gene expression to prioritize the cell type.
That would be also valuable, I think. So you mean separating, so showing the activity of an element across different cell types?
Yeah, because like a lot of gene kind of related to the early point, we don't really know exactly which gene. Like you're looking at this gene expression in different cell type to justify particular GWAS locus significantly in one cell type. But I think if we use the open chromatin region to determine maybe like a better or
no, I think it would be valuable. Yeah. So if you're looking at an element harboring a variant,
right.
What's the activity of that element across different cell types? And I think that's valuable information for sure. I don't know where that goes in the portal, Noel, because there's no like element page, but maybe that's
again, it's just a track right now. I want to get to Alan's question. He's been raising his hand patiently for a while. Alan.
Hi, Kyle.
Hey, Alan.
Have you considered linking out some of these pages to model organism data?
Yeah, maybe Noel. That's maybe more of your.
Yeah. No, this is a great question, Alan. I think you've been a fan of this for a while, which we have kind of put on the lower end of our docket, but we've definitely considered having that incorporated. Yes, the answer is yes, but we haven't done it yet. Do you have any thoughts about how best to do that? Like, what would you like to see?
I have too many thoughts for the amount of time you have here, but this could be a separate conversation because there really is a lot, and a lot of things that are just not really doable in humans it would be really nice to have a quick access to that information.
Yeah, I love that. Yes, we would love to follow up with you, and I will make sure to do that. That's awesome. Thank you. Question from Stephen Gage before we break. Let me just get to the question. Would it be useful to incorporate, since you were working with chromatin accessibility for CREs, and the databases and single cell databases available for long range gene regulatory elements, even interchromosomal, would it be useful to incorporate this into the future, or how difficult would this be to incorporate?
Yeah, I mean, I think that's basically Klaus's question as well.
Yeah.
I think
True.
it's like information on physical relationships between regulatory elements and the genes that they're potentially regulating. And there is a wealth of data. We do have a fair amount of data in this realm. Again, there's many different ways to, and none of which is perfect, to predict the targets of cis regulatory elements. So chromatin interactions, correlation-based things, other types of methods. So we have a fair amount of those data. We don't have any single cell based data, but I don't think that at the moment there's that much like single cell high C type data, just because I don't think there are many labs that can do it. But I presume that will become something that's increasingly common. Cell type resolved interactions based on single cell data. But there's also, like Klaus mentioned, there are cell type interaction maps from sorted cells and also tissue level interactions. So yeah, we tried to aggregate as much of that data as we can, and then yeah, like we were talking about earlier, the way that that's used at the moment is just to allow you to further prioritize variants of interest at a locus with this variant, what's it called, variant sifter page on the portal. But yeah, probably moving forward, there are ways to think about how like you could visualize not only regulatory element activity, but then also putative relationships between regulatory elements and genes and stuff like that in genome browser type settings. But yeah, so to answer your question, we do aggregate that type of data from as many sources as we can find, and it is used to help prioritize variants. But the visualization of it is something that's probably still requires a bit of thought.
Yeah. All right, I'm mindful that we're keeping people a little bit over time, but this was awesome. Kyle, thank you. And thank you for the comments, particularly from folks who actually have always been very helpful in incorporating and giving us great feedback that we incorporate. So I really appreciate everyone's time today. And Kyle, this was a really great overview. And Alan will follow up with you. And Klaus, we need to follow up with you as well. So I appreciate everyone's time. Any other final thoughts? We're going to record this actually, and we'll post in our YouTube channel so folks can see this if they want to reference back to it. And if you have any other questions, feel free to email me directly or our help at kp4cd.org if you want to follow up directly with Kyle. Maybe we can grab him for another session in the future. Thanks, everyone. Have a great day. Thank you, Kyle. Appreciate it.
Thank you.
Thanks. Bye-bye.